So the title of my talk is Synchronicity or Synchrony through Gaze, Mapping the Neuro-Social Network of Infants. So what is the inner brain network life of babies and their parents? So I hope you'll indulge me a little bit because, as I said, there are really two roles for today's seminar. One is to celebrate the science, but also to celebrate how far we've come as a small research group. So this photograph was taken just at the beginning of 2016 when we were just a fledgling little lab in a corner of the Craig Marshall building. And Kylie had just constructed our brand new TARDIS, which we test our babies in. And at that time, you could count very literally on one hand how many people there were in our lab. And since then, in the 2021 months interim, we've essentially doubled in size. We've also eaten a lot more food and drink, celebrated a lot more parties. And I think we've come a long way in our thinking about what this brain-to-brain coupling might be and what it might mean and its relevance potentially for developmental neuroscience. I was also quite surprised and very pleased by this unexpected development, which is that if you go to Google and you type Infant Lab UK into the Google search bar, guess who is the first search response? It's us. So apparently we are the first in the UK for an infant baby lab, and I really apologize to Birkbeck for that. We did not pay Google to do this for us, so temporarily, while you type this into Google bar, the Baby Link Lab is the apparently most highly searched for infant lab in the UK. So I'm very pleased for that as well. All right, so enough of those promotional bits. Why are we interested in synchronicity? Well, synchronicity in my head, in my mind, is an affiliative mechanism, and that's why it's actually useful. So we know that in adults and in groups, synchrony creates social connectedness. And this is not just true for why armies march together in time and bands play together in time and we sing national anthems in time to boost our feelings of patriotism. But it's also interesting in certain case studies as well, which are reviewed very interestingly in this paper by Marsh et al., where they conducted an experiment with two adults, getting each of them to hold a pendulum like this and to try to swing the pendulums in time together. And apparently, the pairs of people who were more successful in synchronizing their swinging behavior, so when the pendulums were more in phase with each other, those pairs, when they then rated each other, also thought that the interaction, the social interaction quality, was more harmonious and more pleasant and more positive, just because they were more successful at swinging something in time together. They did similar experiments with rocking on a rocking chair and other kinds of synchronous behavior, but it seems to be the case that when we act together, when we joint our actions together, that seems to also create social connectedness and a sense of bonding, which is really interesting why that might be the case. And it's not just adults who experience this sense of bonding from synchronous behavior, but infants do so as well. And there was an interesting series of studies that was conducted by Sandra Trehub's group. This paper is Chirelli et al. 2014, where they essentially bounced babies to the beat of music. And so this is the time sequence of a musical piece. And so you had one experimenter who was consistently bouncing the baby in time to the beat. But the baby was watching another experimenter who was either bounced in time to the beat along with the baby, and this was called the synchronous experimenter, or bouncing out of time to the beat with the baby. So this was the asynchronous experimenter. So the baby's there going boing, 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 and the other experimenter is either going up and down at the same time as her or going in a random fashion to her. And then they then asked the question of, does this then support pro-social behavior or helpful behavior in the infants? So this second experimenter then later went into a separate room, and she pretended to drop something on the floor. So she was, hanging up clothes, for example, and she dropped a clothes peg on the floor. And they watched the behavior of the infants to see if any of them would go and help the experimenter to pick up the peg and give it back to her. And lo and behold, they found that experimenters who were bounced synchronously with the babies were more likely to show helping behavior to them afterwards. So apparently it is also the case that from very early on in development, if you act in synchrony with your infant, they're also more likely to be pro-social and positive towards you later. Very interesting and fascinating finding. So inspired by these types of studies, we then began to look more and delve more into the parent-child literature, where actually there is a wealth or an abundance of literature on parent-child synchronicity on many different behavioral levels. So for example, we know that parents and babies synchronize their gaze patterns. So mummies will look at something and babies will follow along, a gaze-following response. We know that they can synchronize their behavior. So if you look at what happens during a period of free play or free interaction, you find that babies and mummies tend to co-fluctuate in terms of whether they're protesting and unhappy or attentive and engaging or playing together. Their states of behavior co-fluctuate together over time. And even closer to the heart or closer to the body, we find that there is co-modulation or co-fluctuation of emotions and even of heart rate and arousal levels. And so you might know of something called stress contagion, and that's a case where the mood of the mother affects the mood of the baby. And so we know that across many different domains of measurement, and indeed many different time scales, Guillaume was talking about this before, that there is interpersonal synchronicity, and certainly this appears to be heightened within the parent-child affiliative bond. So this certainly raises the question, if there's already synchronicity on all these other external levels, is there also the case that brains synchronize to each other? And I'm very pleased I didn't have to make the argument for this, because Guillaume has already showed us that yes, indeed, brains do synchronize with each other, and there's now a growing field of literature examining this phenomena with adults. But do babies also do this with their caregivers? And if they do, the more important point for me is, how is this accomplished, and what purpose does it eventually serve? It's a cool phenomena, but does it mean anything? So in our lab, our general research focus is to look at neurosocial mechanisms of learning, and we are looking at adult-infant neurosynchronicity as one potential example or mechanism by which learning might occur within adult-child dyads. And so when you look at adults and infants together, you can see that they form their own internal close-knit network. So this is a closed-loop network, in a sense, between mum and baby, where babies and mums are locked together in mutual social interaction. And the activities and the actions of the baby are influencing the mummy's brain, and her speech response and her gaze patterns are influencing the baby's brain. And so there's a mutual virtuous cycle that maintains the synchronicity within social interaction. And so you can map it as a social network like this, where we potentially could be talking about the left and the right hemisphere of the adult and the left and right hemisphere of the infant, each sending all possible types and directions of influences to each other during the course of a social interaction. And this two-person neural network is what I'll be describing in the next few slides. So in our lab, we're really interested in studying this state by itself, but we're also interested in what pushes people into the state and out of the state, and also what implications this has for other domains of functioning. So we're interested in what synchronicity cues are there which help two people to synchronize with each other. And so Guillaume already talked about one thing which might help people to synchronize, which is doing things together, so waving or imitating hand motions. But we're interested in things that we know are very common and important in early development, like eye contact and joint attention, and the use of your name, which is a very important and salient cue. When you hear your name, you can't help but orientate towards these. So all of these cues we know are detected by babies very early in life. So might they be potential cues that brains use to synchronize to each other? And more importantly, what benefit does this state hold for cognitive functioning and processing? So are there implications for learning, for communication, and most recently, for development of trust between individuals? And so we're zooming in today just on one of those potential cues, which is gaze or mutual eye contact. And as I said, this is something that develops very early on in life, from birth, effectively. So we know that even from birth, neonates will prefer to look at faces where they perceived direct gaze at them rather than averted gaze. And gaze is perceived as a communicative signal. It's an ostensive signal. It tells babies that you're online, you're ready to talk, and you are addressing them. And it doesn't need any further explanation. It's self-evident. And apparently there's some kind of sensitivity to it even from birth. It's also the case that gaze supports or releases the infant's own communicative impulses as well. So this was a really nice experiment, I thought, that was conducted back in 1975, when you were allowed to submit figures like this. And this is the kind of time plot of a baby, S215, and the number of vocalizations that SIT baby was producing per minute. And there were several different experimental conditions. There was a baseline condition where the adult was essentially ignoring the baby. And then there was two manipulations, one where the adult was giving responses or feedback to the baby, but in an independent, non-contingent manner. And this feedback was smiling and touching the baby. It was dependent in the sense that if the baby said something, then the experimenter would smile or touch. Or it was independent, which means she just smiled and touched anyway, whether or not the baby was vocalizing. And so there were two phases in the experiment there where she was giving either independent non-contingent reinforcing or contingent reinforcing. But the more important manipulation here was that at the same time, she was either offering eye contact or no eye contact to the baby. Okay, so same things being done throughout, but she's either not looking, and they did something quite unusual. They essentially created eggshell glasses for the experimenter, kind of like what we did. And so the experimenter in the eye contact condition was wearing normal glasses. And in the no eye contact condition, they blacked out or they kind of occluded the eye portions of the glasses and just left a pinhole so that the experimenter could see through. So that's how they took out gaze as a factor. And what's very clear in this graph is that whether, when response was independent or dependent, so the contingency really didn't quite matter all that much. What really mattered was the eye contact. When the experimenter was offering eye contact, babies vocalized back more. So from this result, they concluded that gaze has an important communicative function in releasing the infant's own efforts. It tells the baby that you're online, you're receiving, talk back to me now. So the other thing that's important is that longitudinally over development, the ability of the baby to follow someone else's gaze. So if I look over there like that, it usually means that I've seen something interesting and I'm about to, talk about something interesting. The ability of babies to follow your gaze in a different direction apparently is predictive of later language learning ability, specifically vocabulary. And this we know because usually when we turn towards something, we're about to name it. So if I look at that, I'll go, chug. And because I'm following my gaze there and I'm saying something, we can map the relationship between the referent and the label, the referent being the jug and the label being the word jug. And so babies' ability to use gaze and to follow it as a cue predicts their later language ability. So clearly gaze plays a very important role in setting up early cognitive development, and babies are sensitive to it from early on. But more importantly, in the context of our research, is it a synchronizing cue for socially interacting brains? And so I'm going to tell you now about a pair of experiments that we did exactly to test this hypothesis. So here we were looking at the effect of gaze on adult-infant neural synchronicity. Now there's a lot of information packed into this plot, so I'm going to slow down a little bit and let you take this all in. So we conducted two experiments, experiment 1 and experiment 2, with two different cohorts of babies, but the same female experimenter. And in both cases, the babies were about 9 months of age. And in experiment 1, we had 17 pairs and 19 pairs who made it into the analysis. We actually tested close to 30 babies in that one. There was a very high attrition rate from movement and fussiness, et cetera. So this number is how many ended up in the actual analysis database. Okay, so what was the difference between experiment 1 and 2? In both cases, we were looking at brain-to-brain coupling between the adults and the infants. And we were interested in whether gaze would potentially increase coupling between the adult and the infant. But the difference was the modality of presentation. So in experiment 1, the adult wasn't there in person. She was just there on a screen, and her EEG was pre-recorded while she was making the stimuli. In experiment 2, this actual adult was live and present and interacting with the infant. So what was she doing? In both cases, she was singing nursery rhymes to the infant. Twinkle, twinkle, little star, wheels on the bus go round and round. I can't remember what else we have now, but several familiar nursery rhymes of different natures. And then what we did to vary gaze was we had her either look directly at the infant while she was singing, or turn her face to the side and avert her gaze. And so we're going to refer from now on to those two conditions as direct, when she's looking straight ahead at the baby, and indirect, when she's turning her head away at a 20-degree angle and her gaze is also focused away from the baby. And we were interested in, of course, whether synchronicity would be broken when you break gaze. But because experiment 1, we could control the parameters of this experiment much more carefully, we decided to include a third control gaze condition, because we didn't want any of the effects to be because this side face view is just strange. Babies just don't like seeing the side of a face, and that could be why they're reacting differently. So we put a control condition, which we call direct oblique. And in this condition, she was engaging in Mona Lisa gaze, which essentially meant her face was turned sideways, just like the indirect condition, but her eyes were swiveled forward so that she was looking through the corner of the eye directly at the baby. So what we did there was we controlled for the side of the face angle, but she was still offering direct gaze. And that for us was a powerful control, because if you get the same pattern of responding for this one and for this one, it must be about direct gaze. It can't be about the visual parameters of the stimuli. Okay? So that was only the case for experiment 1. In experiment 2, we just did direct and indirect in a live situation. Okay, what did we measure? So we were interested in brain-to-brain coupling. So we measured concurrent EEG between adults and babies. And so we had the infant and the adult facing each other, I mean, virtually or really. And we focused on 2 electrodes, C3 and C4, for each of them. Now, if you download our paper, there's a 20-page appendix which comes with it, which explains why we focused on C3 and C4. In experiment 2, we actually collected 32-channel EEG from each of them, and for experiment 2 was 2-channel EEG. But the main reason is because when we analyzed the pattern of motion artifacts that were generated during speech production, the only place that we could be absolutely sure there was no contamination by motion artifacts was that central region, and specifically in the theta and alpha band only. And so we were going for the conservative approach here. We didn't want any question potentially that any synchronicity could be because babies are chewing in time to the adult or anything like that. So we wanted to exclude any potential confounds there. So we excluded both the electrodes and the frequency regions where we saw any potential motion artifacts. So in all the future analysis, you're going to just see 2 electrodes, C3, C4, and theta and alpha bands only. And that's the reason for it, just being conservative. So what did we compute? So we chose one of the more modern measures of connectivity, which is called general partial directed coherence. And those of you who do a bit of computational neuroscience will know that this comes from the family of Granger causality, which is time series analyses of causality. And it's a very nice measure because not only can we tell who is influencing who, so unlike a correlation where you can't tell who is influencing who, they're just correlated, PDC, which is the abbreviation for this, tells you whether it's adult driving baby or whether it's baby driving adult. So you can actually look specifically at those 2 direction of arrows. And it's also a direct measure, which means that it only includes direct influences and not indirect influences within the network. So we computed GPDC for all possible pairwise connections in this 4-channel network. And we're going to show you the results of network synchronicity as a function of gaze. Okay, it's also very important to rule out any effects of random fluctuations in the environment. So let's say during the experiment itself, there were building works going outside and construction was going on bang, 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 and both their brains were hearing that 2 hertz pulse very frequently. Now that could be a reason why you get synchronicity between brains, which is not a good idea. So we want to find a way of partialing that out or taking that into account. So how we did that was essentially to take the data set from adult and baby and do an internal shuffling within itself so that we break the temporal contingency between one epoch and another. And that gives us a baseline measure of the non-specific connectivity that did not arise from the stimulation itself, but arose maybe from general environmental factors. And as you will see, that baseline measure is really actually quite high. So there is either, whether you want to call it spurious or maybe more accurately non-specific connectivity that occurs at quite a high level. So we know that anything that exceeds the significance threshold is something that's definitely related to the stimuli that was being presented at that particular time. Okay, great. So what were our predictions? Like I said, we had 2 experiments. One was video and one was live. So in the video condition, infants were just observing the adult. There was no way that the adult could have known what the infants were doing. So there's no way that the infant's brains can be influencing the adult's brains. There's only a way for the adult's brains to be influencing the infant's brains. So our first prediction was a sanity check, very important, that in experiment 1, there would be only unidirectional influences, and they would run from adult to infant. If we got anything in the infant to adult quarter, well, that's seriously wrong, or we invented time travel. In experiment 2, where we had face-to-face social interaction, well, now certainly we would have bidirectional influences, so we predict in that case we would have both adult to infant and infant to adult directions of influence. Okay, fair enough. Now, the more important thing is the function of gaze. So we know which connection should survive, but then how should this be modulated by gaze? Well, as I told you earlier, we predicted that there would be higher interpersonal synchronicity during the direct gaze conditions and also during the direct oblique gaze condition, and that these both would exceed the indirect gaze condition. Very clear prediction. So if later I show you this, you will see the mean connectivity values for direct, indirect, and direct oblique. This is the predicted pattern. We want to see a V shape. That's what we want to see. Okay? finally, because I went to all that trouble to tell you about babies vocalizing, this is something we also measured in our data to see what was the functional relevance of direct gaze and whether this had anything at all to do with synchronicity. So our prediction here was that, as before, like in that interesting paper that Bloom published, we should see that babies will be more vocal when the adult is looking at them. But I would say that they'll probably be more vocal only specifically when there's a live interactor, a live experimenter interacting with them. So let's see if we found any of these results. And the good news is, yes, we did. So here's experiment 1, which remember is the video experiment. And here are our 3 gaze conditions again for your benefit: direct gaze, indirect gaze, and direct oblique. Okay, take some time to get used to these network plots now. So here are our 4 nodes again: infant right, infant left, adult right. So this is the adult-infant network. So what we've plotted here is only the connections that survive thresholding. So nothing that's below threshold is plotted. So if it's there, it's significant. So remember our first prediction for experiment 1, we should only see adult to infant. We should not see infant to the adult. Yes, perfect. So all the significant directions of influence or causal arrows run from adult to infant. There's nothing coming back from infant to the adult. So we know the measure works, we are sane. And so what we're plotting here is for the theta band and for the alpha band. And our second prediction is that we should have stronger overall connectivity for direct gaze and direct oblique gaze than for indirect gaze. So now the strength of connectivity here is indicated by the thickness of the arrow. So if you've got a very thick arrow there, that's a very strong driving influence. If you've got a very thin arrow there, that's a very weak driving influence. And what I plotted for you on the right-hand side here now is the summary average of all those arrows averaged into one, the average thickness, if you like, of all those arrows there, there, and there, 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 and there. And what do we predict? More connectivity for direct gaze and direct oblique gaze. And here we have it, our perfect V-shaped pattern, both in theta band and in alpha band. So as predicted, interpersonal connectivity, or the strength of the adult's influence on the adult, is stronger when she's looking directly at them, even when her head's averted, than when her gaze is completely away from the infant. And this is the case across both frequencies. Great, so what now happens when we have a live interaction? Oh, sorry, tick. Well, as predicted, so the second prediction is now that we have a live interaction, we should start seeing some influences of the infant back on the baby. And so now, adult influences on the baby are shown in red, and infant's influences on the adult are shown in blue. And lo and behold, there we have it. We've got now significant influences coming back the other way. Babies are now starting to influence the adult because it's a bidirectional social interaction. So tick again there. We've got a bidirectional network where both parties are concurrently influencing each other's brain activity, which is really nice. Now, did we get our second prediction, which is that in the live scenario, we also have the case that there's stronger connectivity for direct gaze than for indirect gaze? Well, here we have the summary plots for adult to infant arrows. So this is the average of all the red arrows, and this is the average of all the blue arrows for infant to the adult, for theta and for alpha. And yes, as predicted, in both directions, in both bands, influences are stronger when there's direct gaze than indirect gaze. So when the adult is looking at the baby, she has a stronger influence on the baby, and it's also the case that when the adult's looking at the baby, the infant has a stronger influence on the adult as well. So it's bidirectional. So we're very pleased that we found this, but the next question was, so what? And how does it work? So the next question is, what's going on? So what mechanistically might be supporting this kind of mutual synchronicity between brains? Now, we don't think it occurs by magic, of course, and it's most certainly not extrasensory because there's lots of sensory cues going on. So what we think potentially might be happening is that gaze acts as a synchronicity cue that triggers phase resetting so that adults' and infants' oscillation patterns align. So this is a schematic for illustrating how that might work. So during indirect gaze, when the adult's gaze patterns are different from the baby's, their brain waves are essentially oscillating in a non-predictive pattern with respect to each other. So one is not predictable with respect to the other. So if the adult's brain's oscillations are these red curved lines, and the baby's ones are these blue curved lines, then they are not phase-locked or phase-aligned to each other. But what happens when we have gaze is that there is a bit of a jolt, maybe, that causes these two oscillation patterns to mutually align to each other so that they start fluctuating together in time. And basically, this is an extreme case scenario where the exact phase pattern is the same, so it doesn't have to be the case. But basically, one is predictable with respect to the other. Now we know that there's already a precedence for this type of phrase resetting going on in response to sensory cues in the environment, because this is what occurs in response to anything salient, whether it's a speech signal or an alarm that goes off. Your brain phase resets to the sensory stimulus. So our hypothesis here is that gaze is just one more salient cue that your partner gives you, which resets your oscillations. And if both people are exchanging these phase resetting cues during the interaction, it's no surprise that over time, the brain oscillations become more and more aligned or similar to each other. So why is this state interesting or important or functionally relevant? Well, one idea might be that in this aligned or network state, information gets passed between speaker and listener more optimally. Now, why might that be the case? Well, you have to go back then to ask what the function of these oscillations is. And there's a very prominent hypothesis which tells you that these oscillations effectively gate the receptivity of your cortex to new information. Now, it's not possible for us to continuously attend to everything in the world at one time. We can't be continuously attentive. So what the brain does, it creates samples of the environment at very frequent time points. So it samples and samples and samples and samples, and it stitches these together. And these samples of the environment essentially occur when the oscillations are in one particular phase. And when the oscillations are out of that phase, it means that the brain falls into a non-receptive or lower receptivity period, which basically means if you give the brain information during a high receptivity period, let's say, for example, it's this peak of the oscillation here, the brain's much more likely to catch it and encode it than if information was coming during the trough, which we can then say is a low receptivity period. So what's the function of this then for two brains which are aligned to each other? So let's say the adult's brains are the red curves and the baby's brains are the blue curves, and we've got the periods of high receptivity there in the transparent boxes and low receptivity in the shaded boxes. So that's receptive, non-receptive, receptive, non-receptive. You can see here that there's not a perfect alignment between when the adult is ready to speak and when the baby's ready to listen. So quite literally, sometimes the adult's speech is falling on deaf ears because baby's just not ready to listen during that point of time. But conversely, when the oscillations are perfectly aligned, then when the adult is ready to speak, she's at her peak delivery readiness, then the baby's brain is also at its peak receptive period. And so baby's ready to listen when adult's ready to speak. So this alignment in time could potentially facilitate speech communication and understanding. And we already know from other hyperscanning studies from Uri Hassan's group that indeed the strength of coupling between speaker and listener predicts comprehension success. But in our experiment, this is just a theory. So do we have any evidence at all that infants are really more receptive during direct gaze? So here's the sub-analysis that I did, which didn't make it into the final draft of the paper. But here's where we computed the inter-electrode connectivity of just the baby's brain all by itself. So taking out the adult of the equation, we've got 32 channels now for the baby. Let's look at how the electrodes are talking to each other within the baby's brain. Do we have any evidence at all that there's a change in the pattern of connectivity during gaze that seems to suggest more receptivity? So what I've done is I've essentially computed GPDC values for all possible pairwise connections between the 32. I've thresholded them at the top 5%, and only the strongest connections are shown here with the black lines. You might not be able to see this, but the important thing is that because it's a directional measure, this tells you which electrode is influencing which other electrode. Okay? Now remember that our main measures were conducted on C3 and C4, which are these two big black blobs here, and this is the map for direct, indirect, and direct oblique gaze. Now, I just did a very simple thing. I just asked, with respect to C3 and C4, during direct gaze, how many incoming connections are there? And then during indirect gaze, how many incoming connections are there? And then similarly, how many outgoing connections are there that are significant? And just with this extremely simple measure, we can see that in the theta band and in the alpha band, when there is direct gaze or direct oblique gaze, there's much more incoming information to C3 and C4 than during indirect gaze. So it certainly seems to be the case that during direct gaze, C3 and C4 are sinks of information. They're listening much more than during indirect gaze. So some corollary evidence for our hypothesis. Right, let's move on now to vocalizations. So remember, we predicted that under the influence of direct gaze, which releases vocalizations, we should get babies responding back to the adult more frequently. So we coded this, and we used an infraphonological coding system where we measured not just how many vocalizations, but the complexity as well. Was it a marginal syllable, a canonical syllable, et cetera, et cetera. But I'm collapsing all this now into a simplicity diagram of just number of vocalizations and how long they were. Okay, so here we have experiment 1. and experiment 2, so a column for experiment 1, column for experiment 2. And the first row is number of vocalizations, and the second row is duration of vocalizations. And as we predict, during the live interactions, babies were indeed vocalizing more, significantly more to the adult than during indirect gaze. And given that we matched the speech material completely, so the nursery rhymes were identical, the duration of the nursery rhymes was identical, this is completely due to the manipulation of gaze. And we weren't surprised that we didn't see this in the video conditions so much, because obviously that's not a live speaker, so babies maybe didn't feel the need so much to modulate the vocalizations in response to gaze there. So we got a significant effect for number of vocalizations, but not for vocalization duration. So now the other question is, does this have anything to do with brain synchronicity, or is this just an interesting effect? So what we did next was to calculate correlations, potentially, between the strength of the infant's connection to the adult and how frequently and how long they vocalize back to the adult. And lo and behold, we found this. The individual differences in connectivity did indeed matter. So here on a scatter plot, we have all the babies in experiment 2, and on the y-axis, we have how long on average they were speaking to the adult. So how long they cooed or babbled or vocalized back for. And on the x-axis is one simple collapsed measure of just the mean infant to the adult GPDC, so just that bar that I was showing you earlier, just on average. And we found here, lo and behold, a really strong correlation that the stronger the influence of the infant on the adult, the longer the vocalization duration. And it was a very strong correlation that survived Benjamin Hodgberg correction. And it was only the case that this relationship existed during direct gaze. When the adult was looking away, there was no such relationship. So we concluded from this that infants who make a greater effort to communicate seem to be influencing the adult more. And that does make sense, because they're providing stronger sensory synchronizing cues to the adult. But the caveat is, this only works if the adult is looking back and if the adult is actually present, which is not surprising at all. So this actually tells us that infants are active partners in the dyad as well, and they're actually helping to maintain dyadic synchronicity along with the adult, which is quite nice. So one of the last few questions before I close is, we've studied the effect of gaze, we've studied the effect of vocalizations. Are other auditory signals also effective in increasing synchronicity? So we had the opportunity, because it was quite a long experiment, of analyzing the effect of other ostensive cues that were given by the experimenter to the infants, and these were things like saying the baby's name, Tom, when the baby was inattentive, right? Or shaking a rattle. And all of these were very clear bids for the infant's attention. And we were interested in whether these signals would prompt an upregulation or an increase in the synchronicity within the pair. So what we did was we measured synchronicity before and after the signal. So before the experimenter said Tom, and after the experimenter said Tom. And we compared adult to infant synchronicity and infant to adult synchronicity. And as predicted, in both theta and alpha bands, after the adult gave the cue, like Tom or shaking the rattle, there was a significant increase in interpersonal synchronicity within the pair. So this effect isn't specific to gaze. It also holds for auditory ostensive signals as well, when you use someone's name or you shake a rattle to try and attract their attention. However, it was the case that only the adult's influence on the infant increased. The infant's influence on the adult didn't increase, and this was probably because of the time frame that we used. We didn't want contamination of attentional effects. So the after period of time was before the infant had completed re-engagement with the adult, so the infant wasn't yet looking back at the adult here. So we're not surprised that there was no effect here, and there was a stronger effect on this one. Okay, so conclusions, synchronicity through gaze. First, infants can indeed connect to and even help to maintain neurosocial networks with their adults. So they choose their partners, they connect to partners, and they help to maintain synchronicity with their partners. Second, we've shown, I hope quite clearly, that social signals like gaze and using the baby's name can indeed act as neural synchronizing cues between adults and infants. And finally, it could be the case that this type of high synchronicity joint network state may support information transfer between the pair, and in that way may support communication and learning. And it just remains for me to thank all the people in my research team. So thanks very much to Dr. Samuels for dreaming the dream with me, for Elizabeth for being our experimental materials, for Dr. Kylie Claxton and Stanley for helping with the testing. Also, a big shout out of thanks to all our kind volunteers who, as you know, did the hard labor of video coding, hundreds and hundreds of hours of video coding. Thanks to our very brave mums and babies, and of course, many thanks to friends and family for supporting us and for believing in us. Thank you very much. Okay, so I think there's probably time for 1 or 2 questions, and then we'll release you for coffee again. So, anybody? Yes.
Great talk. So I just wanted to tease apart some of those interactions, because we've done some similar studies with NIAs with joint attention, but try to get away from this issue that when there's an indirect gaze, the adult's not looking at anything. And so in the study that we did with NIAs, we had 2 babies wearing the NIA's hat and then communicating with the adults. Every time the direct gaze and singing wasn't to one infant, it was to another one, so it had a reason.
Very good.
So my question is, in that context,
Yes.
if you were then looking at synchronicity, because I didn't do that and we didn't have it on the adult, would you expect there to also be synchronicity with a third partner in a conversation, because the baby is understanding that there's this communication going on in the context of 3 partners, and that there would be something between them? Or in this kind of type of analysis you're doing, it wouldn't? I'm just trying to understand what this synchronicity is telling you.
So there's actually 2 parts to your question. The first is the effects of attention, whether we control that, and the second thing was, if a third party joins, what happens? So I'll address the first one first, which is a really good point, because we didn't want the confound to be that babies were just less interested when the adult was looking away. So I didn't present this, but the infant looking times were identical, at least for experiment 2 for direct and indirect gaze. So they were paying equal amounts of time to the adult. They found them equally interesting, whether they were looking towards or away. So we didn't find any evidence of a drop in attention per se.
It's not a drop in attention, it's an increase in weirdness, because the adult is looking to something and singing, and that's what often happens in babies.
Yes.
We
And that was partly why we did experiment 1 as well, which we had that odd face angle, which looked equally weird. And we also controlled in that condition for what the adult was looking at for her own brain activity. So when she was recording, she saw a paper printout of a baby, and that was following her gaze wherever she was looking. So at least the adult's input was consistent. It wasn't a live baby, unfortunately, because we were trying to keep the cries out of the recording, but she was watching something visually consistent. So in experiment 1, we were better able to control, at least for visual cues, and to rule those out as potential mediators of the gaze effect. So we don't think necessarily it's to do with a drop in attention or a change in attention. But I think to answer your second question, that's a really important point, which is, can infants understand when you need to synchronize with someone else because they are the focus of the information or the cue? And I think potentially, yes, that could be the case. And I would love to see that. We can do it with this measure, because we should be able to see directional switches of the influence depending on who's the recipient or giving the cues. I would really love to do that, maybe a 3-person network, and looking at changes in flow of direction depending on who's the addresser or who's the addressee. So I would predict, yes, you would be able to see that, and you would really want to use PDC as a measure, because then you can measure directional flows from addresser to addressee. So yes, let's do that. In fact, you can do that with NIRS, certainly. It doesn't need to be a fast oscillating time scale signal. Really good questions, thanks.
Can I just have one tiny piece of that, sorry? Which is, because we were also looking at kind of adult to infant, and the other thing that kind of, the reason that we got kind of influence of the infant on the adult in the indirect condition as well, is we were only at 20 degrees, so we put in some pictures. Because 20 degrees, if I'm looking, David, I can still see you quite clearly, and that's about 20 degrees. So that's kind of, because you were just looking at it from the infant's point of view, it's kind of.
Thank you, Sarah. Yes, Claire.
I was wondering about individual differences.
Yes.
So we've got data from our longitudinal study where we started with couples prenatally, where we were asking the parents, the expectant parents, to describe their baby, how they imagined their baby would be. And we've coded that for both coherence, which is used in the attachment literature, but also mind-mindedness. And we're starting to find some predictive links between, especially for the expectant dads, their ability to talk about their unborn child is predictive of their sensitivity at 4 and 14 months. And so I was wondering whether there might be a link between this behavioural measure of mind-mindedness and variability in the extent to which you show this synchronicity, but maybe there isn't enough variability in the synchronicity. So
I
that was the
definitely
question.
share your interest, and that's something we are very interested in with our current data set. So this was a relatively small study with just roughly 20 babies. We have a much larger study that we completed now with 90 mum-baby pairs, where we collected some of that kind of data. So infant's temperament, mummy's personality, home practices, how much face-to-face time do they get. We didn't measure mind-mindedness, which maybe in retrospect we should have, but we certainly do have measures of mom's empathy, etc., along with SES, etc. And the short answer is yes, we predict effects. Because obviously, your home environment is what sensitizes you to these cues in the first place. So potentially it's the case that you only learn to use gaze because you've seen a lot of it at home, and it always predicts something interesting happening after that. But if in your home, your parents never use gaze to interact with you, they use something else, like they shook a rattle. Then maybe you never learned to associate gaze as a socially relevant cue. And so in your case, mind-mindedness might be something that promotes parental practices of that sort. To sensitize the baby to that kind of social cue. So absolutely there would be an effect, we think. We don't completely understand the causal links yet. And I would have loved to analyze that in our data, but we didn't have that background information for this cohort, and also there were only 19 babies. But we did find within that cohort already that there were some babies who were just much more sensitive to gaze than others. So if you do a permutation analysis of just sub-selections of these babies and seeing which are stronger drivers and weaker drivers, there's certainly a subgroup of babies who are much more responsive to gaze than other babies. And it could be the case it's because those babies have a home environment where there's lots of sensitive, contingent, gaze-driven interactions. It could be. We don't know. Yes, really good question. Yeah. Okay, one last question.
Can you talk a little bit about the type of vocalization that you were using during the shared gaze?
Yes, so it's detailed in our appendix, which you're very welcome to download and look through, all 21 pages of it. And there's all this infophonological classification system. So it has 4 classes of vocalizations, and it goes from complexity, essentially. So when it's very poorly formed, it's just basically a long semi-vowel series, like that. So it sounds like a vowel, but it's not quite a fully formed vowel. To class 4, which is fully formed vowel, like duck. CV syllable like duck. And so we've got gradations of complexity there, and based on things like, whether it's clear form and transitions, the duration of the entire syllable, all those kinds of different criteria lets you classify vocalization as being category 1 or category 4. If you look at the appendix, we find that overall there's no difference in complexity of utterances. So the distribution or the shape of those utterances is roughly the same for direct and indirect gaze, but what is different is how many there are overall. Do you have a specific hypothesis that you think that there might be more of one type of utterance than another?
No, I'm just thinking from a musical perspective, I'm thinking about the length of time that you're able to hold attention for. So I'm just interested in exactly what it is that you're doing.
Yes.
You've gone quite fine grained.
Yes, it's very fine grained.
About
Yes.
the length of time attention can be held
Okay.
by using different types of utterance, musical stimuli.
So I don't think there was a correlation between babies' vocalizations and how long they paid attention. So I don't think that was that case. I recall, no. But it wasn't the case. It was a semi-musical vocalization because if you remember, these are sung nursery rhymes, and if you just heard some of them, it did sound like the babies were trying to sing back. So they were cooing along, especially for familiar nursery rhymes like Twinkle, Twinkle, Little Star. You find them going, oh, or something like that. And there was a very clear kind of sung vowel rather than a spoken vowel. So you're very welcome to look at our data. We have promised the journal, which is under revision at, to make the data open access. So where it's anonymizable, where the vocalizations are, we can make that publicly available. Yes, yes. This work is in revision in a big journal right now, and we really hope it will take less than 3 years for publication.